Thank you for inviting me. I'm very pleased to be here, and I'm going to tell you about the work that I'm doing now, which is really thinking about drug discovery from a very different lens in light of what I think is an ongoing revolution that I like to call digital biology. So let me start by talking a little bit about drug discovery, which is, I think, a classic tale of the glass half full and the glass half empty. The half full glass is what you see on this slide, exemplified as a set of different therapeutic interventions across many different diseases that have taken patients that would have had a death sentence or a life of pain and really turned, in some cases, even cured them or at least relegated their disease to one of a relatively straightforward chronic management. And you can talk about, of course, vaccines are very much on everyone's minds now, and rightly so, because I think what we've seen with some of the modern day vaccines is some of the first examples of what is truly an engineered rather than a discovered drug. There is a whole host of biologic therapeutics across different modalities that are exquisitely precise and have taken diseases of cancer and autoimmunity and provided functional cures. And then maybe one of my favorites is just some of the genetically targeted therapies that have taken our understanding of the human genome in diseases such as cystic fibrosis and created diseases that are exactly targeted to specific genetic lesions in our DNA. And in the case of cystic fibrosis, have taken something that was a death sentence at the age of 20 to something that is a relatively manageable chronic disease. So that's the glass half full. The glass half empty is this phenomenon that has come to be called Eroom's law. So all of us, of course, are familiar with Moore's law. Eroom is the inverse of that word. And the reason it's inverted is because unlike Moore's law, which is an exponential increase in productivity, this is an exponential decrease in productivity of pharma R&D for the last 60 plus years, because you can see this graph, which denotes the number of drugs per billion US dollars in R&D spent, approved drugs, is really decreasing exponentially year on year, to the point that the amortized cost per approved drug carrying on its back all of the many failures is about $2.5 billion. And that corresponds to an aggregate R&D success rate of approximately 5%, depending on how you count. So that, of course, is a non-sustainable trend for us to continue on. And what we like to think of doing at In Vitro is to ask ourselves why this is happening. Why is it that the journey of a drug through its discovery and development phases has such high costs and such low success rates? And the reason is because it's a very long journey and has multiple forks in the road. And at each such fork, there is one path, if you're lucky, that will lead you to success, and many, many others that will lead you to failure. And we have very limited ability to predict which is which. And so oftentimes we end up taking the wrong path. It might take months or years and countless millions of dollars to realize that we've taken the wrong path. And so that's what gives rise to the failures and the costs. And so machine learning has become a pretty good tool at this point of making predictions. And can we build machine learning models that will give us at least some inkling, better than our intuitions or heuristics that we use today, of which is the right path to take? Now, no one is aiming for perfection here. Perfection is going to be incredibly hard. Biology is incredibly complex. But can we do better than we're doing today? So the idea that we're looking to build a new kind of drug company that just does this at every step of the way so as to greatly accelerate the process of advancing medicines for patients in need. So why is this the right time to do this? Over the last decade or so, we have been living through two transformative revolutions in two different disciplines. On the left side, you see the revolution that's happening in cell biology and bioengineering, where a whole suite of tools have been developed, each of which is very valuable in its own right, but together they can combine to create a perfect storm of data production. And these involve things like DNA sequencing, what are called induced pluripotent stem cells, which are cells derived from humans, but that can be differentiated with their genetics into multiple different cell lineages, so we can create from one of my skin cells a neuron with my genetics, the ability to perturb cells using techniques like CRISPR so that you can actually explore what happens when you genetically modify a cell, what happens to its behavior. And that behavior can be measured via a slew of different phenotyping modalities that range from super resolution microscopy, RNA-seq, and more. So these, along with many others, are ways in which we can measure and perturb biology in entirely new ways. By doing that, we can create massive amounts of data. And the limitation is really the ability of a human to assimilate those data and to identify the patterns that exist within those that help us identify what happens, what does disease correspond to, and how a treatment might affect things. So the way in which we can address that is using the parallel revolution that many of us are much more familiar with, which is the revolution in machine learning, where in, again, literally about a decade, we have gone across multiple problems from performance that is slightly better than random to performance that is at or beyond that of a human being. And that's on problems where human beings are actually pretty good at, like taking a natural image and creating a caption for it. For problems where people are not so good, like, for example, understanding subtle patterns in cell morphology, or even more so in transcriptional profiles, machines are going to be outperforming humans by a much more considerable margin. So this is a bit of an introduction on machine learning. For many of you, I'm sure this will be familiar, but we're taking a particular angle on it that will really help us understand what we plan to do this in the context, what we plan to do in the context of biology. So on the left hand is the standard depiction of what's come to be called end-to-end training, where the idea is that rather than doing old style machine learning, which is where we had to have a human take any new data set and ask how one represents that in a way that the computer can then create a machine learning model based on that representation. And it turned out, as we discovered, that while using human intuition to create a feature representation of a data domain accelerates the process initially, it also forces it to asymptote at a relatively lower level of performance because people are just not very good at defining predictive features. Today, we don't ask people to do that. We take the data in its raw form, pixels, for example, and images, reads if you're doing sequencing, for example, and the computer builds for itself an increasingly complex hierarchy of features built on top of other features that are all optimized towards an objective, which in many cases is the prediction task that we're looking to solve. And by doing so, the machine constructs a set of features that are much more fine-tuned to solving that problem and able to make very subtle distinctions, like between an Arctic fox and an Eskimo dog, where frankly, a person would be hard put to make that distinction, far less describe how they did that. So that's what's allowed us to create high-performing predictive models in many domains. The other perspective, though, on what's going on here is that the machine is also constructing a novel representation of the data that is optimized towards a particular problem, a particular task. So for example, if you think of images, say, as residing in 1,000 by 1,000 or 1,000,000 dimensional space, the final layer of this neural network is usually about maybe 100 dimensions. So effectively, we've created a 100-dimensional feature representation of the original data. From a mathematical perspective, on the right-hand panel, that is a 100-dimensional manifold that exists in a 1,000,000-dimensional space, which is represented here as a 2-dimensional manifold in a 3-dimensional space. What's important to realize is that the geography of that manifold is very different than the geography of the original space. So distances are very different. For example, those two trucks that you see on the left side of the panel, let me see, I think I can get a pointer. These two trucks in the original representation in pixel space are as different from each other, as distant from each other as any two random images, because they share no similarity at the pixel level in color, shape, texture, or anything else. In the higher level representation that encodes concepts like wheels and windshields, they're very close to each other, as they have to be, because otherwise the machine wouldn't be able to give them the same label. What's maybe less obvious is that objects from other classes, like for instance, cars and tractors, because they share those same high-level features, are also going to be in the same general area of the space, even though the machine doesn't have to give them the same label. And so effectively, we have uncovered an underlying semantic structure in this space that sheds light on some underlying semantics. So how does that translate to our work in biology. So if we think about people as the original basic the original basic objects that we're looking to learn stuff about. And imagine that people are represented by high content data that encodes their biological state. That high content data can be measurements of them as human organisms. It can also be measurements of cells extracted from their genetics and differentiated into appropriate lineages and measured in all sorts of different ways. But either way, we have a very high dimensional representation of a person. And the idea is, can we uncover an embedding of people into a low dimensional space where that geography that we see captures some kind of disease relevant similarity? So in order to do that, we need some sort of labels, and we're going to use genetics of individuals that lead to disease as ways of labeling people as being sick or healthy. And then we're going to basically lay people out on the space and uncover which people are more similar to each other in the context of how, what these biological measurements and the geography that we've uncovered tell us about the disease, the underlying disease processes. So this is very abstract. It will become more clear as we talk about exactly how we do this, but this is the intuition, and this is how machine learning overlays onto the biology problems that we're looking to solve. So in order to address the drug discovery problem, and with this intuition in mind, we have structured the work that we do at In Vitro with along 3 strategic pillars. The first pillar is the understanding of human cohort data. It's what we call machine learning enabled statistical genetics that allows us to deconvolute biological processes, discover targets, and identify coherent patient segments. The second pillar is a parallel one that looks at human cells as the core data generation mechanism. It has some disadvantages in that it's not as close to the human state because it's at the level of cells, but it also has the ability of being an experimental platform in ways that you can't really experiment on humans. So it has that advantage. And then the third pillar has to do with how do you take targets and biological insights that emerge from the from those first 2 pillars and turn them into actual therapeutics in an accelerated, hopefully accelerated and better manner. So that's sort of the 3 pieces, and they all interact with each other in a variety of ways, some of which I'll talk about later. So I'm going to start and basically give a vignette for each of the 3 pillars just to illustrate how we think about each of them. And of course, this is only an example. So the first pillar is really based on the premise that we want to use human beings as a model for human disease, as opposed to trying to use animal models or something that is very different from human biology. And so in order to really understand human biology, one of the, in a way, in a domain when one can't experiment on humans, is to go back to human genetics, because each of us is effectively an experiment of nature, where Mother Nature has perturbed our genetics and given us insight about human clinical outcome of that varies with the genetics that each of us possesses. So how do we use those data? So first of all, do the data exist? And here the answer is fortunately yes. So this is a beautiful Moore's law graph as opposed to Irum's law, because it too is on a logarithmic scale, and it measures the number of human genomes that have been sequenced since the very first human genome in 2001. And you can see that not only is the number of genome sequence growing exponentially, it's growing exponentially twice as fast as Moore's law. And so by 2025, we will have hundreds of millions, if not over 1 billion genomes that have been sequenced. So a lot of genetic data. Genetics on its own is actually surprisingly useful, but genetics with phenotypes is even more useful. So phenotypes have lagged behind genetics in terms of the availability, but there's still a large and growing amount of those available. So one of the favorite examples of, one of my favorite resources in this regard is what's called the UK Biobank. It's a collection of 500,000 people just from the general population, each of which, in addition to genetics, has also a whole slew of phenotypic data that ranges from various demographics and lifestyle factors, multiple different cognitive and functional tests, biomarker data that includes blood and urine, a lot of imaging data from multiple organ systems, including brain MRIs, cardiovascular imaging, ECGs, and more. So it's an incredible amount of phenotypic data that the UK has made available to any qualified researcher. Other organizations, other countries are now creating other biobanks that have similar characteristics. The US has a similar project called All of Us. It's intended to have 1 million people with much more diverse ethnic backgrounds. The UK Biobank is primarily Eurocentric, so it's great that that will become available over the coming years. And there's other countries that are doing similar things. So that's an incredible resource that allows you to align genetics to human phenotypes. And when you do that, because genetics is to a first cut approximation causal of the clinical outcomes, there's some exceptions around the margins, but it's a reasonable way of thinking about this. It allows you to associate genetic variation on the one hand with clinical variation on the other, and assume that the genetics is what's causing the phenotypic change, thereby identifying arguably causal drivers of those phenotypic changes, specifically of disease outcomes. This is not a new discipline, and over the past over 10 years, there has been many papers written on what are called genome-wide associations, where people have looked at genetics, looked at phenotypes, and understood that connection. And there's thousands, if not tens of thousands, of associations that have been, actually tens of thousands, if not more, associations that have been made along those lines. So you would think that we're basically done. We can look at Crohn's disease or Alzheimer's and say, these are the genetic variants that have been associated with those diseases, and those are our drug targets. Now, the challenge, of course, is that pursuing a drug target, as we discussed, is a multi-year, multi-$100 million project. And for each of the diseases that we're talking about, with a few exceptions, there's actually many, many variants that have been associated, often with small effect sizes, at least at the level of the general population. So you can't just say, this is the one that I need to work on. How do you pick, of the hundreds of variants that are associated with each of those diseases, the ones that are worth making an investment in? So this is where having a biological data set that is much closer to the genetics potentially gives us insights about the causal biologies that are enacted, if you will, by the genetic change. So I'm going to give you an example of how of what we did here in the context of the collaboration that we have with Gilead in the area of NASH, which is non-alcoholic steatohepatitis. NASH is a disease of the liver. It's caused by an accumulation of fat, lipid droplets in hepatocyte cells in the liver that over time become inflamed. And when they become inflamed, the cells develop a process called fibrosis, which gives rise to scarring and ultimately to liver cirrhosis, which requires a liver transplant and often, in fact, leads to hepatocellular carcinoma. So NASH is one of the fastest growing diseases, both because of the increasing prevalence of metabolic syndrome and poor diet, and also because it's currently heavily underdiagnosed because it requires a liver biopsy. Up until that point, it's a silent killer. So this is what NASH histopathology looks like, and you can basically see that this is the NAFL, non-alcoholic fatty liver disease stage, which actually is prevalent in 25% of the population, that gives rise to these processes of steatosis, plus inflammation, plus ballooning, and ultimately to fibrosis, which is really the strongest predictor of mortality via liver failure or hepatocellular carcinoma. So this is what NASH is, and what we were trying to address is we had access to a wonderful data set that was obtained via our collaboration with Gilead from a clinical trial that they conducted over 54 weeks with 550 patients. And they were trying to see whether one of their drugs actually had a beneficial effect in terms of slowing or reverting this fibrotic process. The trial did not succeed, the drug didn't work, but it gave an incredibly rich and very well structured, high quality data set. And the question that we looked to ask was, can you uncover genetic drivers of fibrotic progression that might give better drug targets? Now, if you know anything about genetics, you know that there's an awful lot of hypotheses, and 550 patients is actually a relatively small number compared to the multiple hypothesis testing that you have to do. And sure enough, when we looked at that data set, there were really not very many examples of fibrotic progression or regression, and we were simply not powered to see any genetic drivers of these processes. But that's where the intuition that we had was that in these images of these liver biopsies, there was a heck of a lot more information than just the 4 scores that the pathologist assigns to the pathology samples, which I illustrated on the previous slide. So can we do better by digging into those scores using machine learning? So what we did is we took those liver biopsy images and we trained a convolutional neural network to create a much finer grained and more in-depth version of disease state that is higher dimensional. And we trained it using the pathologist scores as an anchor point, but with the goal of creating a more richly structured representation of the disease state. And that gave rise to a much more quantitative notion of what the liver status is, which is then what we used as a clinical and as a phenotype in order to try and identify genetic drivers of progression relative to these now finer grained scores. And so first of all, just to convince you that the machine learned something sensible, these are the predictions from those learned representations of the pathologist scores on a held out test cohort. And this was using a very simple linear model based on that internal state representation. And you can see that first of all, the machine was very well able to learn to predict those scores. And this is, as I said, a separate held out cohort of separate patients from a separate set of clinical sites. The agreement between the pathologist scores and the machine learned scores is commensurate with that of interpathologist agreement. What's also notable is that the representation of the tiles that the computer selected as being predictive of fibrosis using an attention model or predictive of steatosis are very much the ones that a pathologist would pick as well. So the machine, without being told what steatosis was, learned to identify steatosis or fibrosis or ballooning and so on and so forth. That internal representation that the machine learned also ended up being much better correlated with other biological measurements that were taken from these patients that were never given to the machine for training purposes on any patients. So these are blood biomarker scores that are known to be disease relevant, insulin, something called the ELF score, are all very relevant. And you can see that the machine learned scores are much more correlated with those blood biomarkers than the pathologist scores, every single measurement. Similarly, we had gene expression data from RNA sequencing from the liver biopsies on which the images were taken. And again, we see that the machine learned scores are much more highly correlated with the gene expression data than the pathologist scores. But then most important is that we were actually then able to go back to our original goal and identify genetic drivers of fibrotic progression, which I neglected to say is actually the FDA and clinical endpoint that they're looking for is reversal of fibrosis. And you can see that because we don't have these very coarse-grained annotations from the pathologist, rather we have something that is much more quantitative and fine-grained, we had a lot more examples of progression or regression, and we're able to identify 2 novel genome-wide significant variants of fibrotic progression in NASH that, if you actually start looking at them, are very compelling from a biological perspective and eminently prosecutable from a therapeutic intervention. So we've effectively identified 2 very novel drug targets for this disease. Okay, so that's a pillar 1 vignette. Let me move on to pillar 2. And again, doing just an illustration of one of the efforts that we've done along those lines. So this is how we plan to use these cell-based predictive models of human clinical outcomes as a way of understanding and identifying interventions in human disease. So pillar 2 begins where pillar 1 leaves off, with an understanding of the genetic architecture of a disease. That can be obtained in some cases from the literature, and in other cases, it can be obtained from some of the pillar 1 analysis that we talked about. So that genetic architecture is then something that we take and, with all of its richness and all its complexity, mirror that in a population of these induced pluripotent stem cells, which, as I said, are effectively neurons that carry, or hepatocytes or anything that carry the genetics of individuals from which they were taken. So you can create that genetic diversity in a population of iPS cells by either taking them from diverse humans up front, or by engineering additional diversity into those using CRISPR or other technologies to engineer variants that we know to be disease causing or disease modulating. So now you effectively have a population in a dish that mirrors the complexity of the human genetic architecture. You can take those cells and differentiate them into the cell lineage that is most relevant to your disease. So if you're doing a liver disease, it might be hepatocytes. If you're doing a neuronal disease, it might be neurons or microglia or astrocytes. Those cells can further be perturbed using environmental factors that drive a disease state. And then those cells are effectively like a little microcosm of a clinical population in a dish. We can assay those cells using a range of different high content readouts like microscopy or single cell RNA-seq. And with those high content readouts, we can go back and say, for an individual with that genetics, what would their clinical outcome be? And that gives you a training signal that you can then use in order to identify what aspects of a cell state are relevant to healthy versus disease. And so that gives you insights on the patient population, it gives you novel targets, and it gives you a screenable platform for identifying interventions that might modulate the disease state. So in order to make this concrete, I'm going to use a beautiful example from a paper that was published by a group at UCSF in Cell Reports. And I love this example because it's so visual and so clarifying in terms of what we mean here. So there's a region on chromosome 16 that, for whatever reason, is sometimes has a copy number change in humans. So sometimes it's deleted in some people, and in other people, it might be duplicated relative to wild type. And it turns out that when it's deleted, individuals have about a 75% penetrance for autism. And when it's duplicated, they have a 40% penetrance for schizophrenia. And there's multiple genes in that region, and it's very hard to figure out which of them, or perhaps more than one, has that effect on these phenotypes, and what are the underlying biological processes that are implicated in this clinical outcome. So this group from UCSF took cells from people who are deletion, people who have duplication, and the wild type, and they differentiated them into neurons and looked at them under a high resolution microscope. And these are what wild type neurons look like. And this is what the deletion patients look like. And this is what the duplication patients look like. And even to the untrained eye, you can see that in the deletion patients, there is a significant excess of synaptic arborization. The soma are bigger. And there's a lot more synaptic connections. And in duplication, it goes in exactly the opposite direction in what looks like a dose-responsive manner. And sure enough, if you look at this from the perspective of quantitating those phenotypes, you can see that there is a statistically highly significant difference in these phenotypes in these cells. So that is effectively now a screenable platform that you could use for identifying interventions that might modulate this phenotype differently across different individuals. And so this is so now imagine taking something along those lines and applying it in cases where the phenotypes are not so blindingly obvious and where you also want to automate this approach. So the idea is, how do you take very large amounts of high content data of cellular phenotypes and use machine learning to make sense of it? I'm gonna actually skip past this. So in order to do that, what you need is generate very large amounts of cellular data. And so what we've built over the last 2 years at In Vitro is a high throughput automation system that allows you to maintain, differentiate, and measure large numbers of cells. So take the iPS cells out of the freezer, you wake them up, you transfer them to a system we call iClan, which is the In Vitro cell line management system. You let them grow, you passage them, all using robots. And then if you want to perturb them, we put them on this device, which is called the lab site echo, which allows cells to be perturbed in ways that do not involve using a pipette to manipulate them, which is much better for them. This is all using acoustic liquid handling. And then you can take them and put the cells on the microscope and see what they look like, or you can put them on a sequencer and measure their RNA transcriptional profiling. So this is very much a work in progress. What you're seeing are bits and pieces, and there's a lot more to be built to really create that lab of the future, which we're looking to set up, that allows these experiments to be done with very high quality and at very large scale. So what do you do with the data once you get it? And so again, I'm going to give you now a vignette of work that we did also in NASH. That's because that was the first effort that we got into. So we've done the most on it, and there's the most that we can describe. So this was cells that were obtained, in this case, from 9 patients and 9 matched controls. So 9 NASH patients and 9 controls that have a similar BMI, type 2 diabetes status, and so on and so forth. And the question was, can we identify, by looking at these cells, disease versus non-disease, and understand the phenotypes that are involved? When we showed the images from the NASH patients versus the controls to a human biologist, they looked at them and they couldn't really tell the difference. And sorry, I should tell you what you're seeing here. The pink blobs are the nuclei. These are the pink is a stain for the nucleus. The blue is the cell membrane, and the green is a stain for lipid. So these are lipid droplets in the hepatocytes. So people couldn't really tell the difference between the NASH patients and controls. Fortunately, machine learning is much better at identifying subtle patterns than people are. And so you can see on the left a very nice separation between, on the training set, between the NASH patients versus the controls. And on the right side, that same model applied to a separate validation cohort of 3 separate patients and matched controls. And you can see that that separation largely still continues to hold. So the machine has discovered something about the biology of NASH that has allowed it to make the distinction. And the question is what? And so we've also spent a fair bit of time on explainable AI and understanding what it is that is being learned so that a human biologist can interpret it. And so this is just one of the simplest visualizations, but yet very effective, of showing the biologist the highest ranked tiles for NASH versus control. And but it also shows various ways of visualizing what's in the interim. And when you put all those together, it turns out that the difference between the NASH and controls that allows the machine learning to see the difference, and this is also reinforced by attention models, is not the total amount of lipid that you have in the cell. It's not the number of lipid droplets, but rather it's having these large lipid droplets at the nuclear membrane that really is the distinguisher between the NASH and controls. And that is a biological insight that is reinforced by the genetics that we have also uncovered, and gives us new insight about the pathophysiology of NASH. Okay, so that's pillar 2. The last thing I'm going to talk about is very early work that we started to do on machine learning enabled therapeutics design. And the goal here is that we already have a whole slew of interesting targets that are coming off of our biology pipelines, and how does one turn those into actual therapeutic interventions for patients in a way that doesn't require a 3 to 5 year medicinal chemistry campaign? So what we've done here is we've gone to a technology, which is also something that's really emerged as a huge, interesting opportunity in the last few years, which is what's called DNA encoded libraries. So DNA encoded libraries are a very cool idea that allows you to basically create very large chemical libraries in all in a single pooled form. So what it is you take a building block of chemistry, a little piece of a little chemical fragment, and attach it to a DNA tag. So here you can see that they're attached to each other. And then you have a whole bunch of these, let's say 1,000 different chemical fragments, each with a barcode defining its identity. You take those, they're all pooled, you split them into a bunch of wells, and then you add another bunch of fragments, each with its own barcode. And so what ends up happening is that you end up with now these building blocks that are now connected to each other. So these are now larger molecules comprised of 2 fragments and a barcode that denotes that these are the 2 fragments that you have in this well versus these 2 fragments in this well. So if you do this twice with 1,000, say, circles and 1,000 different triangles, then you have a library of 1,000,000. If you do it 3 times, you have a library of 1,000,000,000, all pooled together, each with its own barcode. Now you can take that library, all of which is in this little pool, and you put in your protein target that you're looking to get binders to. And the cool thing is that you now have all of these molecules swimming around, binding to your protein. And when you pull out the protein, say it's on a pole or in a magnetic bead, then you can now say which of the compounds, which of the molecules actually bound to that target. So you've effectively done 1 billion binding experiments in one test tube, which is really cool. And you can deconvolute it, using DNA sequencing. So in most cases, when people apply a DNA encoded library to a particular protein target, what they do is they use it as a substitute to what typically is called high throughput screening, in which you look at the top, 100 most plausible binders, you pull those out, and then you use that as the starting point for your medicinal chemistry campaign. What we do is something very different, which is if we have 1 billion measurements, we should use all 1 billion to build a machine learning model that creates a relationship between the structure of the compound and the binding affinity of the target. So in order to enable that, we actually ended up acquiring a company in October of last year. It's called Haystack Sciences. And what distinguishes Haystack from a lot of other DNA encoded library technologies is that you don't just get fairly coarse-grained dichotomized binding data. You actually get something that looks like a much finer level affinity curve, where this is the affinity as measured by their indexer platform, and this is the binding affinity as measured 1 molecule at a time. And you can see you get very quantitative measurements throughout the spectrum, which gives you much better training data on which you can train a machine learning model that goes from the structure of a molecule to its binding affinity. The important aspect of this model is that you don't use it just on the DNA encoded library. That's not very interesting. You can use it on very large collections of virtual compounds that exist in many cases in the public domain, and identify molecules that are plausibly or predicted to bind to your target in ways. And the nice thing about these molecular collections is that those molecules are much more drug-like than anything that is in your DNA encoded library, and also much more diverse in their structure. And in many cases, those are things that you can go off and buy off the shelf, and within a week you have a collection of molecules that you can try in your assays, whereas often the rate limiting step in your chemical campaign is just the complexity of synthesizing new molecules. And so here you get to short circuit that by focusing on molecules that are already available. So you can put that in, you can you can, so this is sort of the beginning of the process. You can, of course, also put in other properties that you care to select towards, like, for example, various drug-like properties about absorbency or cell permeability. And so you can start to layer on additional predictive models and do a lot of your design work in silico. And before you ever acquire a compound, put them into some of the experimental assays and then go back and do this again, going around this loop again and again. So this is a proof of concept work done by others, not us, but it's a beautiful piece of work that gave us a lot of conviction around this approach that we were taking. So so this is work that was done in a collaboration between folks at Google and folks at a DNA encoded library company called XChem, where they trained these machine learning models on DEL data for 3 different proteins. And what they were able to show is that by screening by screening their machine learning models in these large virtual libraries, they found a pretty good hit rate in terms of compounds that have reasonable, not great, but reasonable binding affinity. But more importantly, they also discovered some number of potent compounds, which are these dark bars in these examples, that are really very high potency and potentially good starting points for the chemical campaign. The other aspect of this, and this is also, it turns out, if you're a chemist, really important, is that you confirmed is that the hits that were obtained are actually quite diverse in their scaffold. So you're not stuck just trying to optimize one molecule. You have multiple options to start with, which avoids getting trapped on trying to fix liabilities in a compound that are going to be really hard to deal with. So maybe just to wrap it all up, in bringing ourselves back to the beginning of the presentation, our goal is to apply machine learning to multiple steps in that process that goes from the very beginning of a drug discovery effort all the way through to the end. So when you think about, for example, our work on target discovery, which happens in Pillar 1 and Pillar 2, we've shown that we're able to identify new targets that, new biologies and new targets that perturb those biologies. We are working on some of the molecular stuff in allowing us to design better drugs and do it faster by doing multi-parameter optimization with machine learning and active learning. And finally, by understanding by understanding the patient population, we are able to segment those patients into coherent subgroups, each of which might have its own characterization genetically and might benefit from a different treatment. And we've already seen that in some of the work that we've done in NASH as well. So maybe just to sort of wrap it all up, let me just take a very big step back and tell you why, after all the many things that I've done in machine learning, why I'm really excited about this particular area. So if you think for a moment, waxing philosophical about the history of science, there are times in history where a particular discipline has all of a sudden made a huge leap forward because of a new technology or a new way of thinking about things, and with a tremendous amount of progress in a very short amount of time. So in the late 1800s, that discipline was chemistry, where the discovery of the periodic table moved us away from alchemy and trying to turn lead into gold into something that looks much more like modern day chemistry. In the early 1900s, that discipline was physics, where we started to understand the connection between energy and matter and between space and time. And then in the 1950s, that discipline was computing. We were suddenly able to generate artifacts, machines that were able to perform computations that up until that point, only people, and sometimes not even people, had been able to perform. In the 1990s, that discipline was data. On the one side, which is related to computing, but also involves elements from statistics, from optimization, from neuroscience. The other discipline that took off at about the same time is what I call quantitative biology, which is the ability to measure biologies at much larger scales and higher fidelity by leveraging techniques that relied very much on advances in physics and chemistry, but also on computing. And that involves ideas like microscopy and sequencing and many, many others. And those two disciplines that emerged kind of in the 1990s evolved in parallel without a lot of back and forth communication between them. And I think that what we're seeing now is the convergence of these two fields into a single discipline that I'm calling digital biology. And that is a discipline that is enabled uh, by the ability to measure biology in remarkable throughput and fidelity, to interpret what we're seeing using ideas from machine learning and data science, and then to go back and engineer biology using various chemical and genetic interventions to do something that it would never otherwise do. This has incredible implications in healthcare, which is what we're doing, but it also has implications in agriculture and biomaterials and in the environment. And I think it's an incredible opportunity to bring those two fields together and have that next epoch of science really transform many aspects of human life. So I think it's a really wonderful field to be in. Thank you very much.
Great, thank you. So we have about 8 minutes for questions. So either feel free to type them into the chat, or you can also raise your hand and I'll call on you from the participants view. So there's already one question in the chat.
Okay.
Should I read it? Okay. Thank you for the insightful talk. Very interesting and inspiring. I'm not very familiar with DELs, and I'm wondering how do you decrease the false positive rates when performing the screening with machine learning models? And how do you ensure the quality of the molecules, such as drug likeness?
Ooh.
Do you use any multi-objective optimizations?
Okay, so that's a wonderful question, and I'm not necessarily the expert on all of these, so I'm going to give a number of different pieces of the answer, and hopefully that will at least help address some of those. So first of all, in terms of addressing false positives, there are a number of controls that one does. So for example, you take the same set of DNA barcodes without the molecules and you run them through the same experiment, and you see whether what binding you get that is due simply to the barcodes and not to the molecules. You can similarly do the same experiment with the DNA and the molecules, but without having the protein on the solid support. And again, you will find things that bind to the matrix that holds a protein in place, but that are not specific to the protein target that you have. So those are some standard controls that you have, in addition, obviously, to some of the more mundane stuff like doing multiple replicates and such that help address some of the false positives and so on. A slightly more sophisticated way of doing not just a control, but to identifying things that are not just binders, but binders to a particular pocket, is to do a competitive experiment in which the protein target is incubated already with its natural ligand, in cases where you actually know the ligand, and then you could actually use this as a way of seeing which of the molecules are binding at the pocket in that they displace the natural ligand that is already there. And so that's another way of doing something that is more of a control. It measures not only binding, but what you might call active binding, or binding at least in the active pocket. So those are some of those controls. Getting the molecules to be more drug-like is a real challenge in DNA-encoded libraries, because the kind of molecules that you can produce using combinatorial synthesis by putting together these building blocks, they're not very pretty molecules in many cases. They're kind of ugly and they're not very drug-like. It's partly because of needing to have them composed of different building blocks, which makes them often larger than you'd like, but also because the kinds of chemistries that you can do on DNA with the DNA barcode is more limited, because the DNA itself is fragile and it needs to be preserved. So some chemical reactions you just can't do with a DNA barcode. And so that's why actually the machine learning on top of the DEL is much more useful, because it doesn't just use the DNA encoded library elements themselves as the starting point for chemistry, but rather uses them as training data. But the stuff that you get from scanning the model against a virtual library can be a virtual library of things that are considerably more drug-like, and much better starting points for chemistry. And that is exactly one of the reasons why this is a really powerful addition to using a DEL, is because you're not forced to stick with just the kinds of molecules that you can put on a DEL.
Great. So then for the next question, I don't know if I'm pronouncing this right, but I think Michal Kazimierski has a hand raised.
Yep, that's right. Hi, thanks for the incredible talk. That was really interesting. So I know that when using cell lines for drug discovery, we often face like the issue of substantial distribution shift between the cell lines and the patients. So is that something you also see with those induced pluripotent stem cells? And if yes, is there any space for interesting machine learning like domain adaptations to solve that?
Yeah, no, that's a fascinating question. And I think, yes, the answer, of course, is that you see, it's not, I would say it's even beyond the drift, because you've effectively, when you're using induced pluripotent stem cells, you've reverted the cells back to their stem cell status, and then you differentiate them to a new, to say a neuron or hepatocyte in a brand new way. So it's clear that there has been a divergence from the primary cells. How do you get insights from these very different looking cells that often are not fully mature, and it's very hard to get them to full maturity in a test tube, and whatever you do also often takes a really long time? How do you do some kind of domain transfer is a really important and fascinating question that definitely could benefit from a lot of novel machine learning methods.
Okay, thank you.
Okay, I think we have time for one more question. I think Petter, you're next.
Hi, thank you for the talk. It's very inspiring. I had a question about this third pillar, and sort of what your opinion is on whether and at what stage it could be possible to also start learning across different targets. Maybe once we have structural information about the targets, whether we can integrate that into a better model.
The answer is absolutely. It's a beautiful question. And I think it's one where up until now, no one has collected enough data to really do that in the context of DELs. There have been some attempts to do that, and I think with some reasonable success in other types of chemical data, like from Chembol, where there's been some attempts to transfer across proteins, and you know, again, reasonable but not overwhelming success, and one can discuss the reasons why. It's definitely the case that one of the reasons we acquired this company and brought it in-house is because there is the feasibility and the aim to automate and scale up some of the workflows so that we can generate enough chemistry data across not just one or a handful of targets, but a much larger number of targets. So you can start thinking about various mechanisms for transfer learning and you know, other ways in which the protein structure itself can be used to help inform the process. It's a great question.